All right. Hey everyone. My name's Jake, and for my semester-long research project, I decided to study amiodarone. I chose this drug because my dad had open heart surgery last summer, and post-op he had to take just a bunch of medication for a long period of time. So when this project was kind of first introduced, I was talking to him about it, and he was like, "Oh, you should just choose one of the drugs I'm taking." And this is the one that I wound up with. It had a good kind of basis of understanding and knowledge, but still a lot of possibility to expand on uh, further research. So this drug is an antiarrhythmic drug. So it is used to treat irregular beating um, in your heart. It does this by blocking potassium, sodium, and calcium channels. This slows down the action potential and kind of helps your heart restabilize and get a constant beat going to properly properly pump blood through your body. Uh, this drug is very effective for treating this issue, but it does have some side effects. It's very toxic to the rest of your body. So this can lead to liver, lung, and thyroid problems. For my research, I specifically focused on uh, the liver toxicity aspect of it. So amiodarone is a pill that you take, um, gets metabolized, and ends up in your bloodstream. It's then kind of filtered through the liver, and this is where it comes into contact with the liver cells. So it's lipophilic, so it's easily able to pass through the cell membrane. Once inside, it can be metabolized by CYP3A4, which is just a digestive metabolic enzyme that can turn amiodarone into desethylamiodarone. However, this doesn't really change the structure of the molecule enough to give it different traits from amiodarone. So that's why kind of this pathway splits. But just so you know, the DEA has the same effects as amiodarone inside the mitochondria. So going off of that, amiodarone can then passively go into the mitochondria, passing the outer mitochondrial membrane into the inner membrane space. From here, it binds really strongly to protons, which is shown here. And once they're bound, it's pretty much permanently bound. There's nothing in your body that can really pull them apart. So once they're bound, they're kind of bound forever. And then from there, there's a proton gradient in the mitochondria. So there's a lot of protons in the inner membrane space, and not as many in the mitochondrial matrix. So amiodarone is able to exploit this and go into the mitochondrial matrix using that membrane potential. And then once it's in there, it ends up stopping the kind of ATP um, making process, which is carried out by the electron transport chain and ATP synthase. So when this is inhibited, um, ATP production comes to a stop and leads to cell death. Now, this kind of is where my research happened. It's not exactly entirely known how amiodarone stops this inhibition of the electron transport chain, but I am led to believe it's due to just the disruption of the proton gradient with the hydrogen binding. So my research question was, we were hoping to better understand how amiodarone specifically interferes with the cell's ability to produce energy, particularly by disrupting the function of the electron transport chain within the mitochondria. Uh, we believe that this dysfunction stems from a disruption in the proton gradient, which is essential for the activity of several key protein complexes involved in cellular energy metabolism. Um, Now, to test this, uh, we proposed an experimental design to affect or to study how the mitochondrial function and energy production in liver cells is disrupted. In doing this, we're going to have three separate liver cell cultures. The first one is going to be our control group, just to make sure that the liver cells are growing correctly, and we'll have a low dosage and a high dosage culture that are exposed to different doses of amiodarone for a set amount of time. Once that time period passes, we're going to use BCECF AM fluorescent dye. This is a pH indicator, so we can expose the intermembrane space and the mitochondrial matrix to this fluorescent dye and be able to determine kind of the different pH levels within the liver cell mitochondria. And then additionally, on top of this, we're going to do O2 oxygen monitoring, which can help us determine the metabolism rate based on oxygen consumption. The expected outcomes of this is that the treated cells will show a reduced proton gradient difference. So what would happen there due to the H plus disruption, we would expect an increase in matrix acidity and a decrease in intermembrane space acidity. And we would also, this disruption would suggest an impaired ATP synthase function. And then our expected trend for the O2 monitoring is that our control group would continue to have a steady O2 consumption because there's nothing hindering the electron transport chain at all. The low dosage, we would expect a moderate decrease in O2 consumption due to the inhibition. And then the high dose, we would expect a very major decrease in oxygen usage or a complete stop of the oxygen consumption rate. So yeah, this is what my research was on, and this would help us determine the effects of amiodarone on uh, liver cell death.